BBC Radio Manchester on a Monday evening. Citizen Manchester LGBT, otherwise known as The Gay Hour, with Claire Mooney, Andrew Edwards, and with me, Ashley Byrne, with you until 10 o'clock this evening. Now, we talked a little bit about this a few weeks ago, about news that an HIV drug could prevent cervical cancer after studies by scientists here in Manchester. We had a bit of a chat about it a few weeks ago, but tonight we're joined by the man behind all this, Dr. Ian Hampson, from Manchester School of Cancer and Enabling Sciences. Tell us more.
What would you like to know?
Well, start at the very beginning.
Okay.
It's a very good place to start, so I'm told.
Okay. Well, basically, we made the observation in 2006. We originally thought that this... to try and sort of explain it, new uses for old drugs is not new. It's quite big news. There's lots of drugs out there that basically have been used for one purpose, and then these are found to actually work for other diseases. For instance, thalidomide. We all know the awful connotations that drug has, but it is now actually turned out to be a really good treatment for multiple myeloma. Which is, remarkable. So we got some results in the lab. I won't bore you with all the scientific minutiae. I'm sure your viewers would find it, really boring. But
listeners even.
Your listeners, yes, your listeners would find it.
I'm sure that imagination. They're picturing something here anyway. I don't know what it is. But there you go.
Unfortunately,
that's because I was on Northwest tonight.
There you go.
Anyway, basically, where was I? Yeah, we found that we came up with the theory that this drug might actually, which is normally used to treat human immunodeficiency virus, it's part of what's called highly active antiretroviral therapy to treat HIV, and we came up with the theory that this drug might actually work against the human papillomavirus, which is the main cause of cervical cancer. About 500,000 cases per annum in the world today are caused by papillomavirus. And we did some initial experiments in 2006 and got some very interesting results which looked favourable, and that was published, and there was a bit of publicity with it then. And we've since gone on, we've been working on it ever since then, and we've now gone on to publish another paper in the same journal, Antiviral Therapy. And that shows quite clearly that the drug is more toxic to papillomavirus infected cells than it is to the normal cells of the cervix, which is really good news because it means the drug has what's called a very good therapeutic index. In other words, it's about as good in a test tube as we can possibly get before we actually try it in people. Now, the other thing is that this drug is already licensed for use in people. You can take it orally, you can take it in quite high quantities, but the concentration that's needed to actually combat papillomavirus at the cervix and other areas in the body that papillomavirus can cause disease is not achieved by taking the drug orally.
Right, okay.
So what it indicates to us is that we will have to apply the drug topically or locally to the affected area, papillomavirus affected area. We need to get about 10 to 15 times the concentration that can be achieved by oral dosing.
Right.
So basically, that's the story. At the moment, we're trying to set up a clinical trial to try this indication to see whether it actually works in people.
Okay, and if it does work, what will the next process be? I mean, presumably a lot of this is down to money and cost and as you say, the drugs themselves are available, but in this form aren't necessarily available, so
yes,
that will take time, won't it?
Absolutely. To get the drug in the right formulation is not cheap. And we have to work with what we've got at the moment. And we're trying to actually set up a clinical trial in Africa, believe it or not, for various reasons. One of them being the availability of the right type of formulation. The right type of formulation of the drug, which is currently only available in under less well-developed countries. So that's one of the reasons that we're actually trying the indication there. And the other reason for it is that of course in less well-developed countries such as the African nations, cervical cancer is still the biggest cancer killer of women in those countries.
What would the clinical trial entail? What's that? How many people? Where's it done?
That's a good question. Clinical trials are usually divided into what's called phase 1, phase 2 and phase 3. Phase 1 is where you look to see whether there's any adverse reactions, whether the drug is tolerated, whether nothing untoward happens. You're not necessarily looking for an effect for the indication that you're looking for. Phase 2 is where you go looking for an effect. Phase 3 is where you go looking for an effect in a big way. Okay. Phase 1 usually involves somewhere between 10 and 30 individuals. Phase 2, about 200. Phase 3, thousands, tens of thousands.
Oh, wow.
With the associated cost implications.
And how long do these various stages go on for before you can? And what we're all really interested in, I guess, is an endpoint which might be, yes, this is really going to be able to be used therapeutically, and when might that be?
Oh, that's right.
Well, that's not a question.
Yeah, I would love it to be. I would love it to be as soon as possible, because I mean it is one of the. I think this is some of the most important work that I've ever been associated with, because papillomavirus, as I said, is a huge problem. It's one of the major causes of human malignancies in the world, and if we could actually come up with a treatment, it would really, really make a difference to so many people throughout the world. So obviously, as soon as possible is the answer. We need to know whether it works or not, because at the moment there is no treatment for this condition other than surgery. That's your option.
I mean, it sounds like such an amazing, move forward in trying to establish a cure. No government money, no United Nations putting anything in?
That's a complex equation, because you see, you've got on the other side of the coin, you've got the vaccines, papillomavirus vaccines, but these are not a treatment, they're a preventative, and they are only really effective if they're given to people before they actually acquire the virus.
Wow.
Okay, so the problem with that is that, of course, you've got millions and millions of women out there who are already infected, and really the vaccine is not that effective against women that are already infected. And the cervical cancer and other, as I said, other cancers, HPV-related cancers, can take 10, 15, 20 years to form. So that's an awful lot of people for an awful lot of time without an effective treatment other than surgery. So this idea, if it works, will be really, we think, quite effective for that kind of individual.
Can I take you back a little bit, because you described, starting off with this idea. I wonder if this works. I'm fascinated by that, because presumably either no one else had made that connection, or if they hadn't managed to demonstrate it. Where does that idea come from? Because without me to be too scientific and convoluted and complicated, but you see where I'm getting at with that.
Okay, it's a difficult one to answer, really. We did some experiments that pointed us towards it in the lab, that this drug might work. But to try and understand about to actually answer that question, you need to understand a little bit about virology and how viruses work.
Oh,
sorry about that.
I was afraid you'd say that.
Don't look at Ash.
I'm really sorry about this. But viruses are remarkable. They're remarkable organisms. Firstly, they cannot grow or survive outside the host. They don't replicate outside the host. They're so-called obligate parasites. And the papillomavirus, just to give you an idea of just what an amazing organism this is, if you were to take all the genetic material in the papillomavirus and you were to compare it to the genetic material in a human cell, okay, not all of it, but just the stuff that's actually made, the business end of the DNA that's in a human cell, and you were to compare the two, and you were to say a human cell is the size of the Empire State's building, which is 1,474 feet high, I think, and you were to say how big would the virus be on the same scale? It's about as thick as the shag pile carpet on the first floor, about 1.5 inches. That illustrates to you just what an amazing feat of engineering it is for the virus that's so small in comparison, with so little information, that takes over something as complex as a human cell with all that information. So how did we actually get there? Getting back to the question. Well, these
what I was really getting at was it a kind of accidental thing? Thinking of Fleming and his, the mould and accidental discovery, or I'm thinking of romance, really, which may have absolutely no place in viral discoveries at all. But
I always say to my students, do you remember the film Dirty Harry? With Clint Eastwood. Do you remember the famous quotation? the famous quotation is, what you've got to ask yourself is, do you feel lucky? I always say this to all my students. It's a kind of rote phrase that I have in the lab, do you feel lucky? And there is quite a lot of element of luck in the lab. You have an idea and you think, I wonder if that'll work. It wasn't quite like that, but that's the kind of, you take your best guess sometimes. And this wasn't quite like that. We had a few ideas. Basically, viruses, as I tried to explain, are remarkable organisms. But because they're so small, many viruses use very similar mechanisms to take over cells. Okay, because they have to, because they're so small, they carry that little information. They have to actually use very similar approaches to take over human cells, which is what papillomavirus does. Well, HIV, we also know, uses some of the same mechanisms that the papillomavirus does. That is basically how we came up with the idea. You see how the logic there. So we thought, okay, let's have a look and see if the anti-HIV drug Lopinavir can affect the papillomavirus. The early indications that we got back in 2006 were it could, and we've since gone on with a Canadian collaborator to show that it targets the right sort of cells. It actually kills HPV-infected cells, whereas the non-HPV-infected cells, it doesn't seem to have that much of an effect on.
It's all interesting stuff, and of course, for our community, the LGBT community, lesbian community in particular, this is a crucial area, because we invariably on this programme do call-outs for people to have cervical smear tests, so they don't call them that anymore, they call them what they call them now? Screenings and things like that. So it's an important area, isn't it, Claire? Extremely important area for people listening.
Well, I think it's fantastic that there is research that goes into this. You never actually hear about very much research. There's a lot of research around breast cancer, but cervical stuff there isn't.
Well,
really.
It's not just cervical. Papillomavirus also has a link to oropharyngeal carcinoma as well. Oral cancer.
Of course, yeah.
And particularly the tonsils. I know that sounds, but it really does. About 30% of tonsillar tumours are caused by papillomavirus. And oropharyngeal cancers, such as tonsillar tumours, are really, really bad to treat. And they have the same sort of relationship to HPV, papillomavirus infection.
See, had you been my science teacher, I could have gone on a completely different route.
Thank you.
Dr. Hampson, thank you very much indeed for coming in to talk to us this evening. That's Dr. Ian Hampson from Manchester School of Cancer and Enabling Sciences. Please keep us up to date with developments, won't you? And let us know how things go, particularly if you manage to get things up and going with regards to any testing and things like that in the future. That'd be fantastic.
You're very welcome. Thank you.
And Dr. Hampson was speaking to us earlier on this evening here at BBC Radio Manchester.